Okay, guys, so just to help us get started with this, I want you to see that we are working on the biology homework notes over section 2.7. In the Manage Back assignment, you'll see that there is a notes packet that you can download, as well as a link to the Google Drive with our PowerPoint presentations. The PowerPoint presentations go in order of where you will see them in the notes packet. So number 1, DNA structure and function. This is the first part of the notes packet, and it matches up with your notes packet. So let's go ahead and get started with that. So one of the things is that we are going to be reviewing DNA and talking about how it replicates and copies itself. And one of the things that allows DNA to do what it does and be so amazing with all of its functionality and how it maintains um, a universal code of life throughout organisms is based on its structure. And so one of the very first things to notice is DNA structure. And this is what Watson and Crick actually predicted for their replication from their model of DNA. So for them to say it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So, haha, stop copying me. So DNA structure allows it to perform its function of storing information in a way that can be copied and passed down to future generations. So in your notes packet, that's already written here. DNA structure allows it to perform its function. So next, what we're going to be looking at is what's this structure and why is it important for DNA to be able to replicate itself? And the very first one is that they have complementary base pairs. And these are only there as a result of the number of spots where hydrogen bonds can occur between the nitrogenous bases. So because of where the hydrogen bonds can occur, so thymine and adenine only gets 2 hydrogen bonds. It's only capable of bonding here and here. So a hydrogen between a nitrogen here and a hydrogen between an oxygen here. And it will not create a third bond here. And that has to do with hydrogen's valence electrons here, but we won't go into too much detail on that. And then when you look at cytosine and guanine, so C and G, they have 3 complementary or 3 hydrogen bond places. This allows for the genetic message to be conserved when each strand undergoes replication. In other words, the daughter strands will have the same sequence of bases as the original parent strands. So because T can't bind with G, it physically doesn't fit, you're never going, or not never, you do get, accidental base pairing, but you are more than likely going to get T binding with A, which allows this message to remain conserved. And so next, so the next part that allows for DNA to replicate itself is its backbone. And this backbone is made up of molecules with strong covalent bonds. So that's between those sugars and phosphates. This allows it to keep each strand of the double helix together. This maintains the order of the bases along the whole chain. Now, if we didn't have strong covalent bonds on the backbone, then the bases could just pop off and go wherever they wanted. And I think as we've learned and will continue to learn, keeping the sanctity of that DNA together and making sure that it's not, sending messages all over the place and sending its base pairs everywhere allows it to keep its universal message. And then finally, the hydrogen bonds between the bases. Why these are important is because they are relatively weak, right? We actually can refer to them as hydrogen attractions because these can break and reform relatively easily. And we can use a really low energy enzyme called helicase to unzip and rezip the DNA during replication. Think about why that's important, and we're going to preview that in just a moment, but by being able to break apart a strand of DNA, you're able to open it up to be read for a larger picture. Okay, so hopefully that was able to fill in some of the information here, and I think some of this will make a little bit more sense when we actually get to the nuts and bolts of DNA replication. Where we're moving on to in your notes packet now is semi-conservative replication. So I'm going to come to the second PowerPoint and semi-conservative replication. If your notes are not color coded, please color code them. And if yours are also with the boxes over the top like this, you may want to move those. It'll make it a little easier. But what happens in DNA replication is that it produces two copies. Each one of them contains one of the original strand and one of the new strand. So when we're looking at DNA and we look at our model of it here, lovely, right? What we're going to be doing, these orange pieces represent, well, these central pieces here represent hydrogen bonds. I realize now they're changing colors, but these hydrogen bonds are ultimately going to be broken apart. And when we break those apart, that is going to reveal the original parent strand's base pairs. And since, for example, G can only bind with C, when we're bringing in new nucleotides to create this process, to create this new strand, only a C can fit in this base pairing rule. So what we're going to see here is that semi-conservative is the most logical way for DNA to replicate itself. There are some alternatives that people have postulated. One alternative is that of conservative replication, and this is where it's going to leave the two original DNA strands together in a double helix and produces a copy composed of two new strands containing all of the new DNA base pairs. This doesn't work out because there's no easy way to read that DNA strand to make a new strand. It's basically just like that new strand just kind of appears. And the other alternative is called dispersive replication. This is where it produces copies of the DNA containing distinct regions of DNA. So like some old, some new. And so what we're going to end up doing is when we're going through semi-conservative replication, we're going to start with a DNA double helix. So in your notes here, just write in this blank area. We're going to draw a original strand of DNA, and we're going to see that what happens is we get a new strand of DNA, represented by light purple here, into our daughter strands, but also the old original strand in dark blue. And then when we go on to create new strands, right, we're going to continually add new strands. And so people were trying to identify, is DNA replication conservative, dispersive, or semi-conservative? I'd encourage you guys on your own to look at this link and open up the video for yourself to get an idea and visualize it. But we're going to go ahead and talk through it here. So the way that Meselson and Stahl used their experiment was looking at bacterial DNA. It's much simpler. It's also not quite as compact. It doesn't have as many histones for it to be wrapped around. You only have one circular DNA aside from plasmids, right? But you get that easy DNA. It's just one copying process. You're not copying a bunch of different chromosomes. So what Meselson and Stahl did is that they allowed many generations of E. coli to divide in the presence of what they call heavy nitrogen. So this is an isotope of nitrogen. So that whenever the DNA copied, all of their DNA will now contain that heavy nitrogen in its base. Okay, so wherever there was a nitrogen atom, it would now have this 15N nitrogen isotope. And so what they did is they ended up spinning that down in a centrifuge, and they identified that the heavy DNA, just as they expected, would go to the bottom. So the oldest DNA, all heavy nitrogen, went down to the bottom. So they started with, here's kind of a table comparing and contrasting. Semi-conservative, conservative, and dispersive all start at the same time. And then after one cycle, they took the bacteria and transferred it to a solution with light nitrogen, so a different nitrogen isotope that had a smaller number of protons and neutrons in it, or sorry, just neutrons, and allowed to divide. And they transferred that lighter nitrogen medium, and they allowed it to replicate. So what do you predict this would look like After one cycle, when we now have a lighter nitrogen isotope added to it, what would it look like for each of them? If we predicted, what we'd see is that if semi-conservative is correct, then we would see a hybrid where we fall in line kind of, I'm going to come back here. We have a little bit of nitrogen 14 and a little bit of nitrogen 15 after one replication cycle. It should fall somewhere in the middle of the centrifuge tube. And that's actually what we saw here after one cycle. It fell into the middle of the centrifuge tube because half of the old DNA, the heavy DNA, was still present, whereas half of the, if it were conservative, we would have seen it splitting up, right? If it was just both old and new. And then in dispersive, we would have seen it kind of spread all over. But what we did end up seeing is we saw this hybrid model where we had half of the nitrogen 14 in place and half of the nitrogen 15. So the DNA sat right in the middle of the centrifuge tube. That indicated to us that the DNA, one of the old strands was present, and one of the new strands with light nitrogen was present. So we know that that can't be conservative. So with cycle 2, they now went about putting it in a replication process. They were allowed to continue to grow with light nitrogen and allowed to divide. So the bacteria are now going through one more division process. We know that the bacteria, whenever it doubles its number, it's only doubled its DNA once, okay? And they went through the division process, and if we predict what it would look like, we'd start to see a little bit more of a spread, right? We'd start to see more strands that are all new, light nitrogen, and then we'd still see those old strands present. And that's what we saw. We saw that the new strands that were after the second cycle, they're only going to have maybe nitrogen 14. Some of them will. Whereas some of them will now still be that hybrid because those old strands are still present. So that's what we saw. So that doesn't allow us to support the hypothesis of dispersive theory. So we did it again. We divided the bacteria. We allowed the bacteria to grow and divide with light nitrogen. And we saw that now there is one layer of hybrid nitrogen heavy and hybrid nitrogen light, and then a lot of it up at the top, indicating that since we've gone through now 2 replication cycles with just light nitrogen medium, we're going to see more light nitrogen present in our sample. And that's, again, what we did definitely see. When spun in a centrifuge, some DNA was in a band above where the 15N band was, and more of the DNA was above that. So if needed, just check out this video and see if that might help you.